Guys. So I'm going to be looking down today because I'm using my iPad for today's presentation because we are going to be learning about Punnett Squares, which is so exciting. And I got to do a lot of writing, so I'm going to be using my Apple Pencil today. So welcome to today's video. We are going to be learning how to complete Punnett squares. So you might remember this from your middle school life science class. If you do, that's fantastic. If not, no worries, because we're going to talk about it today. So we are moving into SB3, which is to obtain, evaluate, and communicate information to analyze how biological traits are passed on to successive generations. And B is our substandard. You should be able to use mathematical models to predict and explain patterns of inheritance. So your goal for today is to use a Punnett square to explain and predict patterns of inheritance. So the first thing we want to do is check out this video on probability. Then we're going to talk about how we can apply that to Punnett squares.
You and a fellow castaway are stranded on a desert island, playing dice for the last banana. You've agreed on these rules. You'll roll 2 dice, and if the biggest number is 1, 2, 3, or 4, player 1 wins. If the biggest number is 5 or 6, player 2 wins. Let's try twice more. Here, player 1 wins. And here, it's player 2. So who do you want to be? At first glance, it may seem like player 1 has the advantage, since she'll win if any one of 4 numbers is the highest. But actually, player 2 has an approximately 56% chance of winning each match. One way to see that is to list all the possible combinations you could get by rolling 2 dice, and then count up the ones that each player wins. These are the possibilities for the yellow die. These are the possibilities for the blue die. Each cell in the chart shows a possible combination when you roll both dice. If you roll a 4 and then a 5, we'll mark a player 2 victory in this cell. A 3 and a 1 gives player 1 a victory here. There are 36 possible combinations, each with exactly the same chance of happening.
Okay, guys. So with probability, we're just looking at what is the chance something is going to happen. So if we look at this Punnett square on this next slide, sorry guys. If we look at this Punnett square here, basically every time we have a Punnett square, whoops. Every time we have a Punnett square, we have 4 boxes. 1, 2, 3, and 4. Those 4 boxes all create one big square. They're all one part of the whole. So it's kind of like fractions almost. So when we're talking about probability, it's just what is the chance that there will be at least one box or 2 boxes that have the same thing. So in this case, we're going to have a 1 out of 4 chance because 1 box out of 4 is capital A, capital A. We're going to have a 1 out of 4 chance to get lowercase a, lowercase a. And then we have, because 2 boxes here have capital A, lowercase a, we have a 2 out of 4 chance to get capital A, lowercase a. And you'll notice 1 plus 2 plus 1 adds up to 4. So it should always add up to our total number of squares that we have within the big square, total number of boxes. So in this case, we've got 4 boxes. Now, there will be some situations later that we'll talk about where there might be more boxes, but right now we're just going to focus on having just 4 boxes. Oops, there we go.
Welcome to Math with Mr. J. So here we are at the introduction to ratios. So we're going to go through the basics of ratios, how we write out ratios, and just talk about what a ratio is. And as you can see, there are 2 numbers on your screen. We have the top problem, ratio problem, with strawberries and oranges, and then the bottom with basketballs and soccer balls. Now, when we're talking ratios, a ratio shows the relationship between different amounts. So we're going to take a look at the quantity of one thing, right, the number of something, compared to the number of something else. So for example, number 1, we have the ratio of strawberries to oranges. So we're going to compare the quantity or number of strawberries to the quantity or number of oranges. And there's a few different ways we can write these ratios out. So let's first start by counting the number of strawberries. 1, 2, 3, 4, 5, 6. So we have 6 strawberries to how many oranges? Well, 1, 2, 3. 6 to 3. So this shows for every 6 strawberries, there are 3 oranges. And again, strawberries has to come first, the 6, because it comes first in our ratio problem, right? Strawberries to oranges. We can also use a colon to represent this ratio. So 6 to 3, or we can use a fraction. Whatever your numerator is, the top number, that's what comes first in your ratio. So strawberries, 6 strawberries to 3 oranges. Now, just like fractions, when it comes to ratios, you want to simplify the ratio if it applies. And here it does. So we need to find the greatest common factor, or what can divide into both 6 and 3 to simplify here. And 3 is a common factor between 6 and 3, and it's the greatest common factor. So let's divide both the 6 and the 3 by 3. Well, 6 divided by 3 is 2, and 3 divided by 3 is 1. So I'll do the colon way here first. Or I could put 2 to 1. Or the fraction, 2 to 1. And let's take a look at our picture over here. And for every 2 strawberries, there is 1 orange. So our simplified ratio matches our picture over there of the strawberries and oranges. Okay, so again, we can use the word 2, a colon, or a fraction. Whatever comes first in your ratio, right, strawberries to oranges up here in the original problem, needs to come first in your ratios when you write out the answer.
All right, guys. So, sorry, I had to make sure that I had my um, mouse over here. So a ratio is just a way of looking at the information that we're given. And so with the image or the not image, the examples he gave with strawberries and oranges, you can do oranges to apples for this picture here. You can use the colon, you can use the fraction, or you can use the word two. But if we look here, let's say we're doing oranges to apples, we would count the number of oranges, 1, 2, 3. That's going to be our first number. And then we count the number of apples, 1, 2, 3, 4, 5. That's our second number. You can do the same thing with pears to apples, or you can even put multiple parts in there. So we can do apples, which we know is 5 already, oranges, which we know is 3, and then we can count up our pears too. That's going to be our last one. And when we do this, your numbers should add up to the whole. So if we add this up, all of them, you're going to get 10. So in that case, 5 plus 3 plus 2 is 10. There's 10 pieces. So when we're looking at these ratios in terms of our Punnett squares, everything should add up to 4 unless we've simplified it. Before it's simplified, it should add up to 4.
Thanks for stopping by. Today we're talking about the basics of Punnett squares. Punnett squares measure the probability or likelihood of a genetic outcome based on a specific genetic cross. It doesn't tell you what will be, only what could be. For the simplest of Punnett squares, you'll cross 2 genotypes. These parent genotypes will be for the same gene but may or may not contain different alleles. Let's look at an example of a genetic cross for eye color. Green eyes being dominant and blue eyes being recessive. And we'll use big G for the maternal genotype and little g for the paternal. So this is our cross. The mother has green eyes and the father has blue eyes. And we want to see the probability that their offspring will have either green or blue eyes. For this cross, we'll draw a square split into 4 quarters, like a simple 4-panel window. This is the Punnett square. One of the parental genotypes will go on top and the other will go on the side. It doesn't matter which genotype goes where, and the order of the letters really doesn't matter, but we conventionally write the dominant allele first, if there is a dominant allele. So your setup should look like this. Then you simply carry the letters through to their respective quadrants and match them up, like so. All right, each quadrant will represent a 25% chance that that genotype will result in the offspring. So in this case, we have only 2 possible genotypes, big G, and little g, and each genotype has a 50% chance of showing up in the offspring. Similarly, you have 2 possible phenotypes, green eyes and blue eyes, with a 50% chance of each. Let's look at another example. We'll cross 2 genes that are heterozygous for eye color in this example. So big G crossed with big G. These colors are just for illustration purposes. So we again will draw our Punnett square and put one gene on top and the other down the side, and then carry the letters through to their respective quadrants and analyze the results. So this cross results in a 1:2 to 1 genotype ratio, with 25% homozygous dominant, or big G, 50% heterozygous, or big G, and 25% homozygous recessive, or little g. The phenotypic ratio would be a 3:1 ratio with 75% green eyes and 25% blue eyes. Okay, hopefully that helps you understand simple Punnett squares a little better. Genetic crosses can be much more complicated, and I'll make a video sometime in the future explaining how to tackle those more complicated genetic crosses. If you have any questions from this video, throw them in the comments, and don't forget to like and subscribe. Thanks for watching. I'll catch you next time.
All right, guys. So that is one of my favorite explanations for how to do Punnett squares, but we're going to work through this Punnett square together. And I'm going to just kind of step by step show you, just like he did, how to set it up. So the first thing you have to do is draw your Punnett square, and it's always going to have 4 blocks on the inside, unless there's more than one thing, more than one gene. But right now we just have one gene. So we're going to go ahead and look through. We've got hornless cattle being dominant. Remember, dominant means that it is going to be the one that's expressed. So that's why it's capital H. It's dominant over having horns, lowercase h. So we have a homozygous hornless bull. So remember, homozygous means 2 of the same. Hornless would be capital H. So that means this parent is going to be capital H, capital H. And it's mated with a homozygous, so again, 2 of the same, horned cow. That's lowercase h. So this parent is going to be lowercase h, lowercase h. So we want to know the genotype and phenotype of the offspring. So we put one parent across the top. And just like he said, it doesn't matter which one is which. I typically, whatever the first parent is in the problem, that's the one I put on the top, but it really doesn't matter as long as you keep them together. So the other parent goes along the side, and then we just carry our information in. So we're going to bring our capital H's down into each box. And then we are going to bring our lowercase h's across into each box. And then we can analyze it. So our genotype, remember, that's our letters. Phenotype is what it looks like. So genotype is the letters, phenotype is the physical traits. So in this case, all 4 boxes are capital H, lowercase h. That means it is a 100% chance of being heterozygous. So remember, hetero means different. So that's where we get capital H, lowercase h. 100% chance of being heterozygous. Now, remember also that if the capital H is present, that means that the dominant trait shows. In this case, the dominant trait is being hornless. So it's going to be 100%, whoops, hornless for the phenotype. Hopefully that makes sense. If not, make sure that you let me know in a live class. And let's look at another problem. So a couple of things. We want to use Punnett squares to help us predict possible outcomes, possible genetic makeups. Just because we do a Punnett square doesn't mean that's always what we're going to see, but it's going to tell us what is the chance of different things. So for instance, we have a problem here. Black fur is dominant to white fur in mice. So we want to cross a homozygous dominant male, that's this one, and a homozygous recessive female, that's this one. So think about what's the probability that the offspring will have black fur? I'm going to toss that question in and let you answer it, and then we're going to see what the answer is. So remember, we're going to bring our top letters down. So capital B, capital B, capital B, capital B, and then we are going to bring our side letters across, lowercase b, lowercase b, lowercase b, lowercase b. So if you got a 100% chance of having black fur, you would be correct. Whoops, went too far. Sorry, guys. So you can see here, this is the Punnett square listed out a little bit neater. It is a 100% chance because every single offspring possibility has a dominant allele. That's because this parent up here, they can only give the dominant allele, which means no matter what the other parent gives, there's at least one, which means that it's going to express the dominant trait. So next, we're going to look at this one. We want to know the probability of the offspring having a yellow seed color or a green seed color. Sorry, excuse me, guys. I thought I was going to sneeze in your ears. Okay, so we can tell from this picture here that yellow is dominant because it's a capital Y. So when we cross, in this case, a yellow seed with a green seed, so we know it's a yellow seed, it shows us here, and a green seed. That means the green seed is recessive because it's two lowercase. When we make that cross, we just count up what we see. So we've got 2 boxes that are yellow, and then we have 2 boxes that are green. Out of 4 total boxes, that means it's going to be a 50-50 chance, 50% chance up here, and then a 50% chance on the bottom one. All right, guys. So you can see here how to complete Punnett squares. You can see how to analyze them, and we're going to talk a lot more in live class about how to do that. So don't hesitate to let me know if you have any questions, and we'll do a lot more practice in class. I hope you guys have the best day ever. Bye.